This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1137, recorded on August 2nd, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 81 degrees and it's raining right now. And it's going to be raining until between 6 and 7. And so I'll either walk home or wait till it dries up because I have my bike here today.
Here in Chelsea, it is 33°C, cloudy, and 60% humidity. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is 92 Fahrenheit, 33°C. They're saying that the humidity is only 44%, but it feels worse than that. And we are under a severe thunderstorm warning until actually for the next 30 minutes. So if I disappear, that's probably why.
We just had a thunder crack, so we've got it right now too.
And from Austin, Texas, Rich Condit.
Howdy. We've got 96 degrees and clear skies. 42% humidity, though, like Alan, it feels like more than that. We have largely dodged the really toxic Austin or Texas weather so far. Had relatively, emphasis on relatively few 100-degree plus days. There's some apparently forecast for next week. We've had an unusually large amount of rain for July, so I'm all for it. It's good.
All right, so that took 5 minutes, the weather, which is kind of mixed in with the introduction. I'm saying that because we got a letter last week from a guy who doesn't want it to be read. He says, I love TWiV. It's too long. You should cut the weather out. Okay, so we're going to say 5 minutes out of 120.
Did you tell him about the experiment that we did?
No, I didn't. I forgot. What was the experiment?
So the experiment was for either 2 or 3 weeks, we just didn't do the weather. We didn't say anything that we weren't doing the weather. We just didn't do it. And listeners wrote in like crazy and said, bring back the weather.
Bring back the weather.
So
Each to his own.
Yes.
yeah, we interpret the experiment results
Each
the way
to
we want.
their own.
You know, the thing is you can fast forward on it. He said actually he rides a bike and he can't do that, blah, blah, blah. I don't know.
Listen, I'm sorry, but I can't do this without the weather. The weather is essential, and I want to know what's going on with you guys. We've just extended the weather conversation.
So the thing is that he says the weather, by the time this is released, is totally irrelevant, and to people in the other parts of the world, it's irrelevant. So why do you have to do it? I said it's just a way of talking.
It's just part of our experience. That's all.
Getting things.
It's
going,
a little introduction and chatting, and before we get to the meat of the episode.
You talk to somebody, you say, how's the weather?
Yeah.
What happened, Kathy? Are you still there?
Oh
gosh.
All right, I'm back.
What happened?
I'm back. Sorry. I just tried to expand my window and then suddenly it said, we're uploading your thing. You're great. I was like, wait, sorry. Does everything seem okay at your end now?
I just don't know what to do when it stops like that. But it uploaded, you said. Okay, we're going to assume it's fine. All right, David, we'll fix it.
Sorry.
Could be.
No.
That will mean that we don't have the weather.
We might have an episode without the weather and without introductions.
Changing a window should be no problem.
Yes. No, I was just trying to stretch it out. I must have hit button.
Yeah, it's okay. Don't worry about it. Things happen. If you like these discussions of science that we do with all the associated paraphernalia, we'd love to have your support. You can go to microbe.tv/contribute. And by the way, when you do contribute, obviously through Patreon and PayPal, you can't do this. But if you sent a check, you could always specify what it's for. If someone wrote in the other day and said, I don't want to support you running around the world. Okay, so write on the check for programming purposes. Then it will just be used for programming. Okay, so that's very easy. When you donate things, you can always, when you get a letter from your university, they give you all these boxes, all the different schools and different things you want, or unrestricted, right? So you could do the same thing if you don't like supporting micro TV travel. Which, by the way, I hear echo now. It's got echo. Does anyone hear echo?
I don't have
Mm-mm.
echo.
I'm not
Nope.
hearing echo.
No.
No.
Nope.
Okay. When I travel, I do podcasts,
Yeah.
and if I don't do podcasts, Micro TV doesn't pay for it. Okay.
And sometimes the places you travel are supporting your travel.
Yes. Many places I go to meetings and they support the travel. And often, if I go to Europe, I try and do several. Like the last time I was in Europe, I did 3 podcasts. So it is efficient. Okay. Microbe.tv/contribute. That's where you would go to help us. And it's U.S. federal tax deductible. A couple of news items. The first, very interesting, an article in the New England Journal of Medicine. Twice yearly lenacapavir, or daily F/TAF, that's emtricitabine tenofovir alphenamide, for HIV prevention in cisgender women. This is a clinical trial done in adolescent girls and young women in South Africa and Uganda. So they got subcutaneous lenacapavir every 26 weeks, or the daily oral FTAF or FTDF, which are 2 previously approved HIV antivirals. The lenacapavir is not approved yet; it's still in clinical trial. 2 things that are cool. First of all, they had over 5,000 participants. No participants receiving twice yearly lenacapavir developed HIV infection. Amazing, right?
Yeah.
But lenacapavir is a capsid inhibitor. It inhibits the assembly of the capsid. Very cool. I didn't know this because it's in development, I guess. So that's cool. We don't have a vaccine, but we do have very good antivirals. And this is preventive, right? Pre-exposure prophylaxis.
Yeah.
And the cool thing is subcutaneous twice a year. You don't have to think about it.
Right.
I think that's very cool.
Mm-hmm.
Yeah, this is a major breakthrough, I think.
And by the way, they did have HIV in the placebo group. Okay, in case you're saying.
And in fact, the equivalent number of HIV with the other drug.
With the daily, right. One of the big problems with pre-exposure prophylaxis is you have to take it every day. And it's hard to keep up with something like that. And there's a lot of social pressure to not take it and yada, yada, yada. Something that's given every 6 months is going to be a lot easier to keep up with.
This is
Absolutely.
pretty remarkable.
Yeah.
Yeah, it's tremendous. Okay, and then from the WHO, we have just released in June 2024 the WHO pandemic plan. It's called Pathogens Prioritization, a scientific framework for epidemic and pandemic research preparedness. Very interesting document, 38 pages, talking about what viruses they want work on, what kinds of work in different places of the world, which ones are important. It's very nice.
One of the things I like about this is that they are not, they've, I don't know if this is just now with this report or if this was a gradual thing that they've been doing for a little while, but previously a lot of these lists have been like this virus and so forth. This is this family of viruses and this family of viruses. And so sarbecoviruses, for example, rather than saying, SARS-CoV-2, which I think is a much better approach because that's kind of how virologists and epidemiologists have been talking about this for a long time. Like, yeah, a novel coronavirus could pop out. A novel influenza of, H5 seems like a reasonable possibility. So I think this approach is better.
At the end, they have a list of the scientists who contributed to this. 1,652 pathogens were examined. This is viruses and bacteria. And so for each viral family, they have all the scientists who contributed. And then they have the advisory committee at the very end. So it's worth checking out. It's very nice. And it's done by region of the world, too, what pathogens are prevalent in different parts of the world.
I really like this because this question comes up all the time.
Yes.
Okay, what do we do? And as a matter of fact, one of the papers that we are doing today, I think, really speaks to this as well, in
Right.
terms of the nitty gritty of if you've identified a potential pathogen, what do you do to figure out how serious a threat this might be?
And lastly, an article in the New York Times, which I can't access, but some of you may. West Nile resurgence in the U.S. Anybody, the mosquitoes are out. Here's, and then the rest of the headline is obscured by.
So I'm not sure if this is a product of my particular set of plugins in Firefox or what, but I have I can toggle the reader view in Firefox,
Oh, yeah, that's right.
and
I
it
can do
takes
that in
that
Safari.
overlay off.
I just put in a gift link too.
Oh, okay. I don't think I have the whole article, but I
Yeah,
am
the
at
reader
least able to
doesn't
get the
offer.
first
Oh,
couple
here you go.
paragraphs.
Oh, mosquitoes are out. Here's what to know about West Nile virus. Infections which can cause fever and fatigue tend to peak in August and September. And talks about current levels. I've only got the first page, yeah. So it talks about the illness and how it's transmitted and the current levels and so forth. So it's a simple, straightforward article. All right, on to the literature. First, a paper in Nature Microbiology. SARS-CoV-2 related bat viruses evade human intrinsic immunity but lack efficient transmission capacity. This comes from a group of scientists at Yale University. And the first author is Mario Pena Hernandez, who I know. He was a grad student with Steve Goff.
Huh.
From Mexico. Spent some time in Steve's lab, and now he's been a postdoc with Akiko Iwasaki, who's on this paper as well. And a number of people here that look familiar to me: Anna Marie Pyle, Brett Lindenbach, David Martinez, Nathan Grubaugh, Akiko Iwasaki, and Craig Weiland. Now, Craig Weiland, do you remember the Tuft cells for norovirus?
Mm-hmm.
So that was Craig, and Craig hired a technician who had sent in a letter to TWiV. So that was many years ago, pre-pandemic.
Mm-hmm.
Anyway.
By the way, the authors who weren't named are also important. Those are just the ones
Yes, of
Vincent
course.
happens to know.
I just happen to know, and I like.
And we have a
to
single
issue
first
it.
author who's Mario, and a single senior author on this. Is correct.
Correct. Yeah. So this is actually also about assessing pandemic potential, which has been a theme in the last number of TWiVs, actually, and particular. So when SARS-CoV-2 was first identified, we didn't have any virus very much like it. RATG13 was the closest, but it was 96% overall genome homology, and not at all close to SARS-1. But since then, people have been sampling a bit and have found SARS-CoV-2 related coronaviruses, and not many of those have been grown in culture, and so they're just genome sequences. And so this paper attempts to make some viruses, some bona fide viruses, and study them. And these are the banal viruses, bat anal specimens.
Yes.
That's where the word comes from. But of course, banal has another meaning in the literature, lacking in originality.
Boring routine. Yes.
Right. But here it's in all caps. And
yes.
I was going to give sort of a summary, but Vincent really just did it. Basically, although there's hundreds of circulating bat coronaviruses, only a few of them have been successfully cultured. And so they make infectious clones for two of them, and then they study those in a variety of ways, and can comment then. We could comment later on their relationship with the immune response and their ability to transmit. So it's really cool. They could recreate these by making infectious clones and then studying them.
And by the way, finding these viruses shows that, SARS-CoV-2 didn't look like anything we'd seen before, but that was just because we hadn't looked. It turns out these things are all around in bats, and these are much more like SARS-CoV-2 than RATG13 or anything else. These are obviously very close relatives.
But still not close enough.
Not close enough. This is not the smoking bat. Yeah.
To be the ancestor, right.
The experiments or the publication that describes the isolation of the viruses is interesting in its own right, and I presume you put this link in here, Vincent, to a episode 809 from 2021, about which I had forgotten entirely, even though I was on it, and I actually looked up the paper
I had the same
myself.
reaction. I was like, wait, I think I
hold.
was on that.
But one of the things I want to emphasize is that these viruses where it came from bats in caves in Laos. And it was a collaboration between the Institut Pasteur in France and I guess a sister or collaborative institution, Institut Pasteur in Laos. Okay, so they did the trapping and sampling the bats, and I presume did a lot of the analysis in France. And one of my reactions to this is that this is exactly the kind of thing that needs to be done for pandemic preparedness, and it requires collaboration and trust. Okay, which is sometimes lacking.
Yeah. By the way, is that
Banals.
thunder coming? I can't tell if it's coming here or if it's over my earbuds from somebody else.
No thunder here.
Okay.
No thunder here.
If there were, I couldn't hear it.
Okay.
It's always muted. And banals, they were described in that paper. By the way, Rich, I was corrected a few years ago by someone from that country, spelled Laos. He said, you don't pronounce the S, it's Lao. But
okay.
The problem is, if you say Lao, nobody knows what you're talking about unless they're from Lao.
Right.
Because everyone uses Laos. So I still use, but Lao is it, yeah. Banals, 3 of them, closest known relatives to SARS-CoV-2. And we were very excited about that paper on TWiV because one of them had been grown in culture. And we said, now you can do experiments. And so here are some experiments to look at basically pandemic potential. Can they replicate in human cells? Can they evade innate immunity? Can they transmit? So we're going to look at those answers to those questions in this paper. Now, these viruses bind ACE2 to get into cells, the same receptor as SARS-CoV-2. And so we have genome sequences of these viruses, right? And they are 97% identical at the nucleotide level across the entire genome. But if you just look at the N-terminal domain of the spike gene, and it's the spike protein, the N-terminal domain that binds ACE2, you have BANAL2 has 52 has 98% amino acid identity, and BANAL236 has 60% only, very different to SARS-CoV-2 NTD. So they synthesize DNA representing these genomes. They make RNA in vitro, and they put it into cells, and out pops viruses. So these viruses grow well, over 10 to the 7th PFU per mil. They're doing plaque assays, which is lovely. And so then they first compare replication of these viruses to the prototype SARS-CoV-2 WA01. And all of them look very similar in viro cells producing ACE2 and TMPRSS2. Now, a viro cell is a kidney cell, so this is not particularly relevant. If they're the same or not the same, I don't know what that would mean.
Right, but
But they go
they
on.
start with a cell that's permissive for SARS-CoV-2
Right.
and see
Yeah,
how well these do.
susceptible and permissive, and
Yeah.
they probably grew the stocks in them as well, so that's fine. But they look at more physiologically relevant models: primary human bronchial epithelial cells grown in an air-liquid interface, so they differentiate into a nice interface, and these viruses all replicate pretty similarly. So right out of bats into human cells, it works
Works right off the
without
bat, yes.
right off the bat.
But before we get too much further into this, because they made these molecular clones, but one of these viruses that they made was one of the ones that had been successfully cultured before. I would have liked to have seen at least one comparison, if they could have gotten it, from that one that had been cultured before with these molecular clones that they made for the 236, BANAL 236, because they did do a little bit of engineering to change restriction enzyme
True.
sites or things like
Yep.
that. And so for the subsequent ones where they put in reporters, the pre-existing one that had been already cultured from the wild, they wouldn't be able to use that as a comparator. But in this first experiment where they're just having made the molecular clones, it would have been nice to compare it to
Yeah, it's a good idea. I agree.
biological isolate.
Mm-hmm. So, right. So those are nasal epithelial cells. Sorry,
Those are
human
bronchial.
bronchial epithelial down lower in the respiratory. Then they say, what about the upper tract, which is the portal of entry for these viruses? In primary human nasal epithelial cells, again cultured at an air-liquid interface, the banal replication is reduced compared with SARS-CoV-2, 100-fold less replication, and at 1,000 to 10,000-fold decrease at 48 hours for 236 and 52 compared to SARS-CoV-2. So they don't seem to reproduce so well in cultures from the upper airway, and could be the same in humans or not, right? We don't know. So that's very interesting. Of course, if it doesn't reproduce well in the upper tract, it may not transmit well, right? So that's interesting. They look to see if these viruses are susceptible to antiviral drugs against SARS-CoV-2, remdesivir and nirmatrelvir, and they're all susceptible to these drugs. They take sera from people who are either infected with SARS-CoV-2 or vaccinated, and they are neutralized by sera from these people. So
those were a couple of really important experiments to do.
Yeah,
right
that one really struck me.
Yeah.
Both the drugs, drugs weren't as much of a surprise because those drugs hit pretty generic replication targets. Okay, but the antibody one really struck me, and in terms of this idea of these are the sorts of experiments you might do to evaluate the pandemic potential of something, that's a really significant result. That you might actually have a little cross reactivity from the SARS or a coronavirus that's already out there.
And from a lab safety perspective, it's important to do these right away when you're isolating a virus that could be a spillover.
They next look at the requirement for host proteases for reproduction. Now, SARS-CoV-2 requires the cell surface protease TMPRSS2. Can also, if there's no TMPRSS2 in the surface, it can also enter via endocytosis, and the spike can be cleaved in the endosome by a cathepsin protease. But they find that all right, so first of all, I have to tell you that these banal viruses, the spike does not have a furin cleavage site, right? And so without a furin cleavage site, you have less dependence on the cell surface TMPRSS2. Anyway, there is a cathepsin inhibitor that they first used. So cathepsins would be the protease in the endosome. So if you do an experiment in cells lacking TMPRSS, the viruses enter through the endosome, and you could see the effect of a cathepsin inhibitor, and they have similar susceptibility to SARS-CoV-2 for that. And then they use cells that have temperase 2 on the surface. And then you can use a temperase 2 inhibitor and ask the effect. And there they say the banals have a little bit higher dependence on temperase than does SARS-CoV-2. But basically, like SARS-CoV-2, banals can get in either at the surface or within endosomes, 2 different proteases. Then they look at evasion of immune responses. They look at interferon first. Can these banal viruses counteract interferon? They seem to have enhanced resistance to type 1 interferon. If you treat cells with interferon, they grow better than SARS-CoV-2, in fact, in interferon-treated cells. They equally induce interferon-stimulated genes as SARS-CoV-2. And then if you pre-treat cells with interferon, these banals have higher titers compared to SARS-CoV-2. So they have greater resistance to interferons than SARS-CoV-2. Now, MHC1 is another immune component they look at. MHC1 is a surface protein involved in antigen presentation for CD8 T cell recognition and killing of infected cells. And we know that SARS-CoV-2 downregulates MHC1 so that it can prevent infected cells from being lysed. And interestingly, the banals also downregulate MHC1. They say even though they've not seen humans before, these banals, they can still downregulate human MHC1.
So they must be seeing some conserved pathway.
Yeah,
Yeah.
I assume, yeah.
So at this point in the paper, it actually, other than the nasal turbinate data, it actually looks like these would be pretty good candidates for a spillover.
Right. So far.
So far, yes. This is stuff that you would look for, and if you were, what's the pandemic potential of this? And up to this point, you'd be saying, yeah, we ought to be a little concerned about these.
So then the next series of experiments are in animals, in animal models. What about the pathogenicity and transmission? So first, ACE2 transgenic mice. They infect them, look for weight loss and mortality. SARS-CoV-2, a lot of weight loss, 100% mortality. Banal 236, 15% weight loss, 20% mortality. And no weight loss or mortality with Banal 52. Also, much less viral production in these mice compared to SARS-CoV-2. And so these viruses have a range of pathogenicity. So SARS-CoV-2 is the highest, Banal 236 is intermediate, and Banal 52 is low pathogenicity. In the mouse, do these viruses have resistance to interferon? They treat mice with interferon alpha, and then they challenge, and then they find, like they find in cell culture, they're less susceptible to interferon. They look at virus replication in the nasal cavity. So SARS-CoV-2 you can find at similar titers in the lungs and the nasal turbinates, but the banals are decreased in the nasal turbinates compared to the lungs. So again, kind of consistent with those human nasal epithelial cell culture results. And then next, transmission. They use Syrian golden hamsters to look at transmission. Syrian hamsters get moderate disease, but they can also transmit the virus, and they infect them with the banals or the SARS-CoV-2 viruses. In this hamster model, they get some disease and weight loss with SARS-CoV-2, of course, then followed by Banal 236. And 52 doesn't cause any weight loss, but the viral loads in the oropharynx are lower and delayed for the banals compared to SARS-CoV-2. So then they look at transmission. So this is a contact model. So they infect a hamster and put it in a cage with another hamster, right? So if you want to look at an airborne transmission model, you'd have to put them in separate cages, but they don't do that. And so if you use SARS-CoV-2, all the contact hamsters get infected, but BANAL 52 or 236, none of them get infected. And even if you put them in the same cage. And interestingly, they wonder if this is dependent on how much virus you add. And in fact, they find that you need a lot more banal viruses to infect the hamster, and that's possibly why you have reduced transmission, because you don't get a lot of inoculum, presumably, as much as you can load into a hamster in the laboratory.
Didn't they? I thought there was one experiment where they got one transmission,
Mm-hmm.
right?
Yeah, there's
Yeah, I was.
one dot in figure 4G,
Yeah, it was
and they
one.
talk about it too.
Yeah.
Okay, so the last experiment is they take the furin cleavage site out of SARS-CoV-2 and ask in hamsters what happens. And in fact, it's now has impaired replication like the banal viruses do. So they're thinking that the furin cleavage site is really important for that. And when they do a co-infection experiment with SARS-CoV-2, is it a co-infection or just an infection experiment? Yes. So they infected hamsters with SARS-CoV-2 either with or without the furin cleavage site. But the hamsters infected with the virus lacking the furin cleavage site, a virus outgrows the population which has the furin cleavage site, because apparently their inoculum is mixed. It has some viruses with furin cleavage site and some without. And those with the furin cleavage site outgrow the ones lacking it, which shows that the furin cleavage site is important. It's kind of a inadvertent competition experiment, right? So, you can't just look at replication in bronchial and nasal epithelial cells. You have to go beyond that. And that's what they do in this paper. They look at, immune antagonism and also pathogenicity in animal models.
And this also implies, doesn't prove it, but it suggests that the reason SARS-CoV-2 has a furin cleavage site is because if it didn't, it wouldn't have become a pandemic. Maybe.
Yeah, they suggest that it's important for replication in the upper tract,
right?
And that's important for transmission.
I mean, these are basically, like if you have SARS-CoV-2 without the furin cleavage site, you don't get maybe spread from human to human.
Yeah.
Boy, wouldn't it be nice to have some sort of model that would more reliably predict what might happen in humans. I mean, the ultimate model is humans. Okay.
Yeah.
But throughout this, I'm thinking mice lie.
Yes.
Monkeys exaggerate. I don't know what hamsters do.
Well, these culture systems that they're using, not the viros, but the bronchial model that has an interface and the nasal turbinate model, these are part of this whole genre of more complex culture systems that we're getting with organoids and so forth. And those are, I think, the systems that I would look to potentially maybe at some point in the future even replace, and in some experiments you can already replace the animals with these models, and get better data about what's going to happen in humans. And in this experiment, or this paper, you're already seeing something where the nasal turbinate and the bronchial culture systems were pretty good predictors of what's going to go on in the animals.
Right. And in a way, the bottom line is: are these relative to the known human pathogen? Are these viruses the same, or are they different?
Yeah.
Okay. And they're different, but there's a lot more information than that. I mean, it is striking that you put them into human cells and they grow. Okay.
Yeah. So they make a very nice statement, which I like, is that it shows that viral so there's a transmission defect, right? Yet the viruses did grow in human respiratory tract cells. So they say replication in vitro is not enough to predict efficient transmission. That's really important. I like that very much. The other thing they say is that the lack of the furin cleavage site in the banals may explain their inefficient replication in the nasal epithelium and their poor transmission and poor pathogenesis. But they point out that, SARS-1 does also lacks a furin cleavage site. So it's not just the furin cleavage site. There's probably other things that play a role in transmission and pathogenesis.
Well, and SARS-1 transmits very poorly.
It does transmit poorly.
Compared to SARS-2. And it seems to me that these banal coronaviruses probably could spill over into humans and maybe have, and they just don't really catch on.
At any rate, if you're doing surveillance of coronaviruses for potential spillover as potential pandemic pathogens, one of the things that can be on your radar is furin cleavage site.
Yes,
we already knew that, but
we knew
more.
that. But this paper,
but there's more.
Shows
more
that there's more.
That.
Yeah.
But the experiment you would like to do is to take the banals and put a furin cleavage site into them. But good luck in getting that experiment approved in any place, right? And they say, due to biosafety concerns, we did not do this experiment because it would never be approved, and probably rightfully so.
Yeah.
Right? Even though you could do it under high containment, you probably shouldn't do that. Yeah.
This paper was really nicely written.
Mm-hmm.
Every section has a nice heading that describes what the conclusion is going to be. And then at the end of each paragraph, it concludes that, having shown you the data. It was just so easy and clear to read. I really enjoyed it.
Unfortunately, it's paywalled, as is the other paper we're going to be talking about today.
It's unfortunate. All right. Next paper is Science Translational Medicine. It's all about dengue. A PRM mutation that attenuates dengue virus replication in human cells enhances midgut infection in mosquitoes. This is from a group in Singapore. And we have Choi Sirifanichatkorn, Choi Ui, Manuel Tan Zin Yap, Gubler, and Ui. So the senior author is Ung Yang Ui, and the first author is Alison Choi. I'm pronouncing it wrong. He did teach me how to pronounce it. He was on TWiV when I visited Singapore. That's TWiV 633. And just a lovely guy. Wonderful. If you look at the picture on the TWiV, you can see that everybody's very happy. Everybody's smiling and laughing. And Ui is the second from the right in that photograph. And so I was very pleased to see this paper, which is all about dengue, as it says there, but how it changed during its progression throughout the Pacific. So remember, there are 4 types of dengue virus. They're spread by mosquitoes. We know that population immunity plays a big role in susceptibility, but I'm sure there are viral genetic factors that do also, and this paper tries to address that. So a little history here, which is very interesting. There was an outbreak in the South Pacific Islands in the 1970s. So it was mostly absent from the South Pacific Islands for the previous 30 years. And then in the 70s, there was an outbreak. The dengue 2 strain emerged in 71 in Fiji and Tahiti, and then moved to New Caledonia, Niue, and American Samoa in 72, and finally reached Tonga in late 1973. Now, all these islands except Tonga and Samoa had explosive epidemics with severe dengue, but few cases were detected in Tonga, but they did find virus there from patients who had mild fever of unknown origin.
So something changed between Tahiti and Tonga.
Could be
Maybe.
that something changed.
And it's
This one-
interesting if you look at the map in the supplementary figures. It's
Right.
not like you have a wave going from one place to another, which I suppose, in a collection of islands you wouldn't necessarily expect. The variant viruses show up in spots, in particular in Tahiti. I want to spend a minute here to emphasize the dengue cycle in humans and mosquitoes, just to make sure that everybody understands this. The virus replicates in both humans and mosquitoes, and the transmission is that a mosquito, if we start with an infected human, a mosquito takes a blood meal from a human and takes up the virus, and the virus actually replicates in the mosquito, winds up in the salivary glands, so that when the mosquito bites another human, the human can be infected and replicate dengue, and the cycle repeats. My understanding is that humans are the primary vertebrate host, though other primates and other animals can serve as a host as well. And it's always struck me as, it's not like I know all the details or anything, but mosquitoes are pretty different than humans. And it always strikes me as a remarkable trick for a virus to be able to replicate into such different hosts. And we're going to see the impact of that in this paper.
So the sequence of the genomes of isolates from these islands show that the Tonga isolates have 3 amino acids that distinguish them. An amino acid change in a protein, non-structural protein 4A, NS4A, a change in the pre-membrane protein, PRM, that's the protein in the title, and what's the other one? 3.
It's the one that they don't see even in all of the Tonga
NS2A.
viruses.
Yeah, so not all the Tongas have this NS2A changed. So the purpose of this paper is to ask, what did these changes, the amino acid changes, have any potential impact on what happened epidemiologically here? They think the PRM is the driver, and it looks like that from the findings here. And what happens is, as Rich said, somehow this single amino acid change enhances reproduction in the mosquito midgut. So that's something that would be selected, right? And it's just by chance it happens to reduce fitness in humans, and therefore you have less replication in disease.
And yet those two balance each other out so that it can still persist
It can
in
still
the
persist.
environment.
Yeah.
You get enough replication in mosquitoes so that it compensates for a reduced fitness in the humans.
Yes. So even though it has
and you
reduced
still get
fitness-
enough replication in the humans that it
Exactly.
can get into the next
Right.
mosquito.
Exactly right. Okay, so they have infectious clones of these genomes, just like we saw in the previous paper. And they have infectious clones of the Tonga isolates versus the other islands, and they do plaque assays.
They actually show plaques.
They show plaques, and they have a plaque phenotype. I loved this.
Mm-hmm.
This is classic virology.
It makes your heart go pitter patter.
And not only do they
Yes.
show plaques, but the Tonga isolates make small plaques compared to the other islands, the other South Pacific islands. I mean, that's just gravy.
This is
right?
this is early 20th century virology, finding new things.
You hope you get this kind of phenotype because this is very clear, and you can study it. So not only small plaque phenotypes, but the Tongas have a lower virus reproduction in a human hepatoma cell line. They also have reduced expression of interferon-stimulated genes.
I want to make a point here because this was fascinating to me. They did this by qPCR, I think. Yeah, based on the And what they used as their control was TBP, the Tatabox binding protein. I have never seen that used as a control in qPCR before. I just found that fascinating.
It's cool. Yeah.
Is that, I saw it on the y-axis, TBP, and I was like, wait, that can't be the Tatabox binding protein. And yes, it is. So it's cool.
So lower replication, reduced innate induction by Tonga, right? And they say this is different from what we saw with a vaccine strain, PDK53, which is part of the tetravalent infectious attenuated dengue vaccine TAC3, which replicated more rapidly than wild-type parent. But for that virus, this vaccine component, the interferon response limited replication of that virus. So they wanted to say, make sure that the reduced replication of Tonga is not just a matter of interferon inhibiting it. And so they they actually infect mice lacking type 1 and type 2 interferon receptors. And so that means they don't have an interferon response. And the Tonga strains still replicate more poorly than the New Caledonian strains, which shows that the interferon is not explaining the plaque size. Or suggests, they say. They should have done a plaque experiment with interferon.
Yes.
Null cells, but maybe they couldn't do it. But that's the way to do it. So what changed? The 3 amino acid changes in the Tonga genome compared to the others, right? So they make the changes in their infectious clones. And to my delight, they write infection of cells with dengue 2 containing a single PRM H86R substitution. They use the word substitution, not mutation. I was so happy. But
these are
one
TWiV
sentence
listeners.
later, but one sentence later, they then say the PRM R86H mutation.
You can't win them all.
And I know that I'm a pest about this, but they got it right for a bit. Anyway, if you put in this H86R that's in the PRM of Tonga, make small plaques. And if you take Tonga and reverse the change, it makes the plaques bigger again. You're in heaven.
This is also classical virology, and in fact, the making the reversion back and getting the large plaques is what I call the Lois-Miller experiment. So they would make mutations in their baculoviruses, and then to be able to show that the phenotype was due to just the mutation they introduced, they would make a revertant, and then they would compare the revertant to the wild type, and it would be comparable if everything was right.
I noticed the same thing. I had the same reaction, Kathy. I didn't know that it was the Lois-Miller experiment.
Well, it's what I call it.
I have my own nightmares with that experiment being required. Nowadays, is it sufficient just to sequence the clone you have? I mean, that goes back to a time when sequencing was not as easy to do. So if you made a mutation, you didn't know if you by accident created something somewhere else, and that was the purpose of doing the reversion experiment, show that you can change it back. But I wonder if nowadays just sequencing what you have is sufficient.
So I'm going to find out with this paper we're going to submit, because we have never been able to easily make viruses and to make revertants. And so our approach was to try and isolate a sibling clone. And if the 2 sibling clones behaved the same, that had the amino acid changes.
Yeah, do it twice.
But we couldn't even get that.
Oh.
So we did a whole genome sequencing.
Okay.
So, and we're going to see.
Well, if I'm reviewer number 3, I'm going to say that's okay.
Okay. Thank you.
You're welcome.
Maybe that can be arranged. Doubt it.
So reversing the change in Tonga not only makes larger plaques, but it you get increased replication in mice compared to the other mutation. So this single amino acid change H86R and PRM it seems to explain some of the changes, some of the phenotypes of the Tonga dengue 2. Remember, this is dengue serotype 2. So this change, this amino acid 86, is right next to a furin cleavage site. We can't escape furin cleavage sites today, can we? And so they think, well, maybe the change affects cleavage. If furin cleavage is important for cleaving PRM from the rest of the membrane protein. And in the mature virions, it's cleaved and gone. And so maybe they think, well, maybe this change affects that cleavage. So they do some biochemistry to they do immunoblotting, Western blots, and measuring the ratio of PRM to E. And they found that maturation is not compromised by this change to at position 86, at amino acid 86. What else do they do here? So what they do find is interesting. They say when we look at infected cells by Western blot analysis, so we can see the levels of the proteins, there's reduced production of a number of proteins. And so they think that this amino acid change somehow reduces the efficiency of protein translation in mammalian cells only, not in mosquito cells. They actually look at that in mosquito cells in culture. The protein production is the same with or without that change, but it's specific for mammalian cells. And they have some ideas about how that happens, but they don't really know how it does that in the end, basically.
They do try.
They do try.
They try. I mean, but one of the leading hypotheses is it's codon usage thing, right?
Yeah, right.
And because the codon for this amino acid is different from what is believed to be preferred in human cells, that's thought to decrease the efficiency of translation.
Right.
But it's really hard to nail that down.
Yeah, they said it was technically difficult.
Yeah,
because they would make the changes and they would revert
They
and
would
so
revert.
forth. So they really couldn't look at it. But then they turned to the mosquito and look at the fitness of these viruses in the vector. These are Aedes aegypti, by the way. There they find differences in infection rates and production of infectious virus with Tonga versus the New Caledonian strains. Okay, so they give mosquitoes a blood meal, so it's simulating what would happen when they pick up blood from an infected patient, and they find that for New Caledonia, even the lowest amount of virus, 10 PFU of Tonga, produces 100% infection rates of the mosquitoes, but New Caledonia only 20% infection rate. And so then they get 3 logs lower infectious progeny with the New Caledonian versus Tonga. So New Caledonian strains have a fitness problem in mosquitoes, even though they can still transmit, right? They're apparently not as fit as the Tonga, which is really much better in this experimental model. And they find that this difference is specific to the midgut of the mosquito. Because if they put the virus intrathoracically, then the two viruses have comparable infection rates. So something's going on in the midgut that favors Tonga reproduction.
So, time out here.
Yeah.
I want you to picture doing an
I
intrathoracic
did.
infection
Yes.
of mosquitoes. I looked it up, and there's a methods paper that you can get on how to do this. And as you might imagine, it involves micro capillaries and micro manipulators and all of this kind of, and looking through a dissection scope. So you knock out mosquitoes by chilling them, all right, and then with this apparatus actually inject them specifically into the thorax. My God.
and the mosquito has to survive this in order to
Yeah.
do the experiment.
And finally, they infect mosquitoes, both in a colony established from wild Aedes aegypti and also laboratory passage Aedes aegypti with all these viruses. And they find that consistently the dengue 2 with PRM H86R amino acid change infects the mosquito midgut more effectively than the other viruses. And so more mosquitoes are infected.
That's
after
really
10
a
days.
dedicated experiment. Do you suppose that that's reviewer 3 stepping in there and saying, wait a minute, your colony is homogeneous. What about in the wild?
Because it's the last paragraph of the results, right?
Yeah.
Nevertheless, good
So
on
this
you.
is very cool. This is a very
cool.
neat thing that you have when you have two hosts, a mosquito and a mammalian host, you can have different selection pressures in each one. So in this case, most likely a variant of dengue 2 was selected in mosquitoes for greater replication and so forth. And that results in lower fitness in human cells just by accident. So, if people identify a virus that causes mild disease in people, they always say, oh, it mutated to become less pathogenic because that's good for the virus. And no, it's not the explanation.
It could be good for the virus, but what the virus is doing, what you're doing is you're selecting for viruses that are better at propagating themselves.
In
that
mosquitoes and another host,
yeah, it
right?
could be. And the thing with, say, a respiratory disease is the decreasing pathogenesis often will increase transmissibility because somebody's more likely to be out breathing lung juice on other people when they're not feeling bad.
Well,
with a vector-borne, like especially mosquito-borne disease, the person could be unconscious, and in fact, that'll improve spread because now they're not swatting the mosquitoes away. So you seldom see this sort of pattern, but as Vincent said, this is a case where because it's replicating in two different hosts, there are competing selective pressures.
Yeah. So what I want you to understand is that when you have two different hosts, you can have an effect in one cause something else in the other, whereas the selection wasn't in the human in this case; it was actually in the mosquito, right? So was this? They say this codon change was selected because of the advantage in the mosquito midgut. And then they talk about actually how it might work, but they don't have any good evidence for that.
I would say that this also, I mean, in my mind at least, fits into what for me is today's arc on pandemic potential, because we're looking at virus evolution in real time here, and seeing how, there are different effects on different hosts and et cetera.
Yeah, they have a very nice concluding sentence. Our findings point to the importance of understanding dengue virus genetic factors that may underpin dengue epidemiology.
Yeah. It's cool.
Yeah.
Yeah, really neat paper.
Very nice. I'm trying to think if he talked about this, but it was probably too long ago. That TWiV was recorded in 2019. And what's that? What about
Singapore viral was June 30th, 2020.
Well, I recorded in December 2019.
Okay.
But I didn't release it right away because something happened in January.
Oh, is that right?
Yeah.
Yeah, I had also at the same time in Singapore recorded an interview with Peter Daszak, right?
Yes.
Where he talked about how they were doing these experiments in Wuhan, and people said, he admitted to making the pandemic virus. And then a reporter called me from Fox News once and said, you recorded this in December. Why did you release it in May? Were we trying to hide something? Yep. It's called the pandemic.
Yeah.
Anyway, that's very, very nice principles in both papers, right? Not just the results, but principles that it's not just a furin cleavage site. In vitro replication doesn't tell you about transmission. And you can have selection in one host that impacts the other just by accident. In fact, it's amazing that many viruses can grow well in both a vector and a mammalian host, right? Because you can see one change, one amino acid could mess it all up. Okay, let's do some email. We can probably get through all of these today. I'll take the first one because it's short, because I'll leave the long one for other people. This is Marcus, who writes, when you were discussing spiders and whether or not they eat roaches, yes, baby roaches get clobbered. Spider joke. That spider you just killed for being in your apartment thought you were his roommate. Way to go. Love your show, MJR.
Oh, by the way, Rich, I did the spider experiment with the flashlight. Were you,
The
were you
one
one
that
who
I
mentioned
talked
this?
about.
Oh,
Yeah.
that was you, Kathy. Yeah.
Yes.
Hold the flashlight next to your eye and the
Mhm.
spider eyes glow. Unfortunately, my spiders don't do that.
Oh.
I have a great source for them. My cellar hatch out back. There's an interior door, and then the cellar hatch, and of course I never go in that space, and it's just full of spider webs, and so I went in there with a flashlight. Oh, cool! No, can't see.
So tell me again how you do this.
You hold the flashlight up beside your eye, pointing. I mean, at eye level, but pointing away from you,
Right.
Okay.
toward spiders.
Like the helmets they use in Alien.
Right. Yes.
Okay.
Yeah, but I guess maybe it's dependent on the eye size of the spiders or something.
I
or-
don't know, but the times when I've tried it, I've seen it. But I have
Alright.
to say that it was in Georgia both times. I can't remember if I've tried it in Michigan. So I'll
Okay.
have to do that.
Yeah, I did determine that I have a UV flashlight as well.
Mhm.
And I found the spiders that I have at least are not UV fluorescent, but the pill bugs, the isopods, are.
Oh, cool.
Yeah.
So I'm going to hold this up to my phone.
I see
from
a
my
blurry
phone.
spider. Yeah, there it is.
Yeah, that was on my desk yesterday. It's about
Oh.
Cool.
3/4 of an inch. And like Vincent, I try to save them, but I would have had to go down 2 flights of stairs. I would have had to have something to capture it in right here in my office, and I didn't. And sorry, squish.
Oh, well.
Huh.
The other night when I was doing the live stream, a spider just came down in front of me on a thread, right in front of me. Right, so I picked it up and put it on the side of the
Kathy,
desk.
that spider thought you were his roommate.
Yeah, exactly. So I have to tell you, this is really a digression, but Choral Union, we're sitting on the Big Hill Auditorium stage during one of the recitatives during the Messiah. One of the soloists is singing, and I happen to look up, and it's probably a height of, I don't know, 40 or 50 feet maybe from where we are to the ceiling. And I see in the light this thin line that's a spider thread, and the spider is dropping down, and I see it come down, and I'm just watching it and thinking, there's nobody else that's going to be able to see this. I can't tell anybody. And then it just crawled back up.
Huh.
So it was pretty fun.
Little Miss Muffet.
Yeah,
Yes.
exactly.
Can you take the next one, Alan?
Yes, Matt writes, in your response to the email by Peter in TWiV 1133, you claim there is no evidence that TWiV was working on SARS-CoV-2, which is true. But it's very different from your subsequent claim that you know what viruses TWiV was working on from their publications, and they weren't working on SARS-CoV-2. In some countries, it is not uncommon for institutes to be involved in both civilian and military research. It's also common that publications require prior institute approval. We can judge what an institute is working on by its publications, but this does not automatically mean we see the complete picture. An associated claim that I've heard from TWiV is that the absence of a whistleblower lends support to the virus not originating from a lab accident. This is nonsense. Zhang Yongzhen ended up without a lab because he released the genomic sequence without authorization. Humiliating country and party by a whistleblower on, for example, a military project would likely be viewed as treasonous, and the consequences much worse than for Zhang. The above two arguments also fail to consider large cultural differences, and by association weaken the sensible scientific arguments that you make. Regards Matt, who says he's a researcher at an East Asian institute who has worked on military projects in past jobs and has for the past decade been unable to publish without explicit upper-level authorization. Okay, Matt, couple of comments. First of all, yes, in some countries it is not uncommon for institutes to be involved in both civilian and military research, such as the United States. It is very common for American universities to be involved in both types of research in many different fields. Some places publications require prior institute approval. Fine. This idea that Zhang Yongzhen ended up without a lab because he released the genomic sequence is hogwash. If you actually read the news articles and do a little fact checking on this, it is pretty clear that Zhang Yongzhen ended up between labs because of a dispute with his institution about how the lab would be moved or where it would be moved or something along those lines. It reads exactly like the kinds of things that happen at American universities, and he was camping out in front of the institute as a matter of protest. That was not the guy got kicked out because he said something 3 years ago. Let's see. Fail to consider large cultural differences. Yeah, we should all be afraid of China. Right. I get it. But realistically, I do like your first point that there is no evidence that WIV was working on SARS-CoV-2. That is true. There isn't any. And absent such evidence, you can't go around, or you shouldn't go around, making wild accusations when there's no evidence behind it. And by the way, we just talked about some stuff today that further undercuts the notion that SARS-CoV-2 came from a lab.
I don't understand why you would bring all this stuff in when there's no evidence for any of it.
You
right?
would bring all this stuff in because there's no evidence for it.
I see.
So it's a tactic. I actually have used and taught this to debate teams, how to win an argument, how to win a debate when you have bad arguments. You throw a lot of them out there, and after a while they start to sound credible because there's just so much. But when you evaluate them individually, you find out that they all lack substance.
This other thing here that TWiV said, an absence of a whistleblower lends support to the virus not originating. I don't remember saying
I don't recall
that.
saying
I don't
that.
remember that.
I don't either.
Okay. Kathy, can you take the next one?
Yes, Charles writes, hello, Twivers. 89 Fahrenheit, 32°C. Looks like it could rain, but the forecast says we will stay dry for the rest of the day in Spotswood, New Jersey. This email was prompted by a letter read on TWiV 1131 from Greg. Warning, I am sarcastic by nature, and this email cries out for sarcasm. But sarcasm does not work well in emails, so anything you see in this email that is written in italic and bold should be taken as sarcasm. Okay, so that's this first part right coming up. Dr. J. Bhattacharya, specialty unknown to me, but from his papers he must be an epidemiologist, and Dr. Vinay Prasad, oncologist, are two of my three favorite Bay Area doctors, the third one being Dr. Scott W. Atlas, radiologist. Dr. Bhattacharya for his excellent work on the Great Barrington Declaration. Dr. Atlas for his incredible advice to President Trump during the COVID-19 pandemic. Dr. Prasad for making McGill University's What's Trending in the World of Pseudoscience, and that's link 1. Trend number 6, anti-vaccination to be specific. Okay, so now this next part is not italic and bold, so it should not be sarcasm. Okay, I'm sure you remember Dr. Bhattacharya from the Great Barrington Declaration. You may not know of some of his other work. I first noticed him when I saw a news article about a preprint paper he had written. The paper, COVID-19 antibody seroprevalence in Santa Clara County, California, link 2. This paper should be required reading in every entry-level epidemiology class. Note, I did not use the sarcasm font. The paper drew my attention because it made two extraordinary claims. The first claim was that 2.76% of the county had been infected with SARS-CoV-2 by late March of 2020. That seemed high to me. The second claim was that the infection fatality rate was 0.17%. That was out of line with most other studies at the time that were close to 1%. Following Carl Sagan's advice, extraordinary claims require extraordinary evidence. I looked through the paper looking for the evidence and how the paper could mess up and produce such an extraordinary result. I found the latter, but not the former. So that is, he found the evidence for...
found the error, but not the evidence.
Not the evidence, right. I found this in the paper. We recruited participants by placing targeted advertisements on Facebook aimed at residents of Santa Clara County. That told me all I needed to know. Who's more likely to volunteer for a study like this? A person that has been sick over the last 3 months or a person that has not? I think the answer is obvious. A prime example of selection bias if I've ever seen one. Not trusting my own analysis. Bold and italics coming up. After all, I am just a computer programmer. No more bold and italics. I searched for qualified people that agreed with me. I found an article by Balaji Srinivasan, peer review of COVID-19 antibody seroprevalence in Santa Clara County, link 3. There are others, but I like this one. I did not find any that agreed with Dr. Bhattacharya. This paper ended up published in the International Journal of Epidemiology. That never should have happened. I do not understand why people pay any attention to this person. Not sure if you've ever heard of Dr. Prasad, but I assure you the anti-vaxxers have. I have two big issues with Dr. Prasad and his views of vaccine for young people and how he came to those views. He treats myocarditis induced by vaccines and those induced by SARS-CoV-2 infection as equal. That is not my understanding. I think Dr. Griffin would agree with me. With this view, he does not believe that the mRNA vaccines should be given to young people. He also thinks that testing for the N protein antibodies at this point in time is still valid for determining if a person has been infected with SARS-CoV-2. The problem with this is that the antibodies fade with time, and vaccinated people tend to produce S protein antibodies at a much lower rate, link 4. I don't disagree with everything he says, but I do not agree with his main points. Others agree with me, link 5. End of this long rant. Thanks, Charles. And then here at the end of his letter, he's got all the links that he just referred to. So you can check out all these letters online and look at his points and his references.
I think that people have suggested having Bhattacharya and Prasad and on TWiV. And I said no because these guys have
Don't
said
give them airtime.
not going to give them airtime.
No.
No, and this just underscores that. We don't want people that do things that aren't correct. Yeah.
I have one little subtlety here, just out of for general interest. He talks about Bhattacharya's paper saying that the infection fatality rate was 0.17%. That being out of line with most other studies at the time that were close to 1%. And I have to wonder, infection fatality rate is different than case fatality rate. We went through this early on.
Right.
And I wonder if there is not some vocabulary mishap there, because not all of the infections result in cases. So the infection fatality rate is likely to be much, much less than the case fatality rate. Now, globally, my understanding is that the case fatality rate was all over the map. But I think, the average is somewhere between like 0.5 and 1%. I'm thinking closer to 0.5%. I don't know what the infection fatality rate was. There's going to be at least 50% of the infections that were asymptomatic, I think, and probably more than that. So the infection fatality rate is probably much less than that.
And since we weren't testing early in the pandemic, we had no information on infection fatality rate.
Right. And, unless you're really ambitious, you're not going to get those data because, you get data from people who show up sick. And the people who never knew they were infected, you have to go out and find them if you're going to get that number.
CFR is influenced by so many things, right? Like hospital care, whether the hospitals are overcrowded, whether people know how to treat you, what your age is, your
diagnostic
sex,
capacity. Yeah.
So many things.
Yep.
So many things cannot be used as any reflection of the virus itself, which people tried to do at the beginning of the pandemic. Rich, you haven't read. Timothy's next, please.
Timothy writes, "Hello, TWiV, longtime listener. I work at a small cancer center in Ontario, Canada, where a lot of our patients are treated with various immunotherapies. There actually is a monoclonal antibody, leronlimab, targeted against the CCR5 receptor, that's a co-receptor for HIV, which may eventually be used to treat metastatic breast cancer," gives a reference, as CCR5 is highly expressed in CCR5 is expressed in a high percentage of metastatic breast cancer cells, another reference. There's actually a lot of promise for leronlimab's use in treating triple-negative breast cancer, another reference. though not yet approved in Canada. Just wanted to point this out. Thank you for all your hard work and bringing science to the masses. Kind regards, Timothy. Okay, thank you, Timothy. That's very interesting. Where do we must have been discussing this at some point? I missed out on that. A monoclonal antibody targeted against CCR5 receptor.
I don't remember, but it may be just monoclonal therapy in general,
Okay.
inspired this email. But it's very interesting, yeah. All right, our last one is from Peter. Vincent, I've been thinking of the last decade, more or less, of listening. The essence of TWiV and others was your podcast can go for hours where you talk gibberish. Thanks to someone on the call, it could be any of you, that is the joy. I know a hawk from a handsaw. Then someone chimes in to explain to the rest of us what that means. And that is part of the beauty of what you do. Many thanks, Pete. P.S. I think you should capture as much of Dixon as you can, but you are already doing that.
So somebody appreciates the randomness of this conversation.
Yes.
Yes.
I think many people do. It's just not everyone vocalizes.
People have different degrees of patience for waiting for the random moment that they need.
I think a lot of the appeal of something like this is kind of like watching a series on TV. You get to know the people, okay, and the personalities, and it's fun to watch them interact and that kind of stuff. That's a significant part of the appeal. I mean, that's why I'm here, because you guys are so weird.
And we're not even in Austin.
That's right.
Yeah, that's what I'm here for.
All right, let's do
Everything
some
else
picks.
is so banal.
Right.
Banal. Can we use banal in the title somehow?
Oh, yeah,
I
we'll
have
figure
a
that
suggestion up there for that.
Would be nice. Let's do some picks. Kathy, what do you have for us?
I picked a short video that's put out by Michigan Engineering that is talking about using big data to improve traffic light timing. So it turns out that there's all kinds of data from cars and other things about timing traffic lights, but it's so expensive to retime a traffic light that it only happens, I think it was once every 5 years on average. And if they could make use of this information and take this big data and employ this model and this research that this particular professor has been doing, they could potentially retime traffic lights maybe as often as every 3 months for now, and how that would improve traffic immensely. And they have some statistics. I rewatched it yesterday, so I'm not going to try and misquote them to you, but basically they reduced the number of times that cars had to stop at traffic lights, and they increased the flow in a nearby community in Michigan by using this modeling and these big data sets. I just thought it was really cool. And they show the filming of the traffic areas and stuff, but they speed it up. So everything's moving really quickly. It's just really fun to watch. But
this is really neat. I saw this in one of my news feeds too, and thought, wow, that's really cool. And I saw the Michigan link. I was like, I wonder if Kathy's going to pick this. So I made the right call. When I was in college, in Towson, Maryland, at the north end of Baltimore, and I lived south of Baltimore, I discovered that if you went, I don't know if this is still the case, but in Baltimore, if you went up Charles Street at, I think it was just a little under 35 miles an hour, at least on my Datsun speedometer, if there was no traffic, like it was late at night, you could hit all the lights green. They had actually timed them, but it was only timed for that speed with no traffic. And I don't know how often they had to redo it, but it did work.
Yeah.
And this would make that a lot easier to set
If
up.
I'm not mistaken, when I was growing up, Van Ness Avenue in San Francisco was my father called it the Grüne Welle, the green wave. So if you drove the speed limit on that and got it right, you could get through the whole thing, which is a lot of lights, and they were all green.
Yeah, I remember highways like that. You had to time it right. Oh, here in New York City, of course, it used to be. I don't know if it still is. When you're going up and down an avenue,
Yes,
you could time it and get your greens going all the way and never have to stop.
So
But
I just sent this link to two experts of my acquaintance. One is a guy I sing with who is a retired traffic engineer to whom I complain constantly about all the problems, and I blame him for all of them. And the other is my wife, who is an expert on traffic signals, okay, and intersections. And there's a driving around with her, there is a running commentary on how all of this could be done better. So she'll be interested in this.
So in many places you have short blocks, and then the traffic doesn't work right because they don't have this kind of coordination, right?
Yeah.
So you fill up the block, and then the light is green, but then the one behind you is red. So you never relieve and it should be computerized to take care of that, right?
Yep.
Sure, but
Traffic circles, man.
Yeah,
We need more
Massachusetts
traffic
is
circles.
Massachusetts is massively into traffic circles.
Even
Traffic circles
even
are
if they're
great,
not more
yeah.
efficient, and I think they probably are more efficient, at least you feel like you're getting somewhere.
Yes.
Okay.
Traffic circles are great. Yes, I really like that. Yep.
For those of you in the UK, those are the same as roundabouts.
Roundabouts,
Roundabouts.
yes.
Right.
Siri still calls them roundabouts, even with the American accent.
Rich, what do you have for us?
Okay, I have a recent local discovery called it's recent for me, called Dillo Dirt. So I went to my local mega hardware garden store recently looking for top dressing for my lawn, because my lawn had I'm adamant about my lawn. There's not much of it, which is okay, but it's I don't know what it is, but I dig it. At any rate, there were a couple of lame spots, and so I wanted to give them some top dressing. It's always hard to find good stuff for top dressing. It really works right. And I spotted this stuff that was really cheap. That's a local product called Dillo Dirt. Dillo as in armadillo. Okay. And then I looked into it. It was dirt cheap. I looked into it. It worked great. I spread it out. It was nice consistency. The lawn loved it. I looked into it, and it is a product of recycling, local recycling of wastewater plant solids and compost. So, and it turns out that
Poop and leaves.
the wastewater plant processing facility is 40 years old. This was introduced in the mid 80s. And I don't understand all of it. You can sort through the links here and learn about it. But I have to go visit this place. It's on 1,200 acres in sort of northeast-ish Austin, as I understand it. And it collects wastewater solids from several different wastewater facilities around and goes through a process that matures it, if you like, and also pasteurizes it. And just it involves big million-gallon tanks and stuff. And of interest, a couple of things struck me about it. The heat generated as this stuff ferments, if you like, serves to pasteurize it. Okay, so it gets so hot that it kills all the bugs that are doing all the action. Okay, which is an interesting concept. The other thing is that it generates a lot of methane. They capture the methane and use it to generate electricity. All right, and some of the byproducts of this, that part of this facility is a bunch of farmland that is leased out to farmers, and they can spread some of the byproducts of this on the farmland to help grow wheat, and they sell the hay.
Nice.
To help the facility. So So the other thing is that Austin has a composting program. So we have green bins that we put out, and there's a remarkable variety of things that can go in the green bins. And that's mixed with, it includes, your yard waste, but also, all your leftovers from dinner, okay, pizza boxes, you name it. tissues, used tissues, the stuff that you'd like to put into your regular recycling, but it's too dirty. Okay. So there's also, matter from around Austin that's officially generated, stuff from their tree trimming and et cetera. And I'm sure a lot of other compostable stuff. And they have huge composting facilities. And so part of that compost goes to this wastewater solids treatment plant and gets mixed with the wastewater solids and composted further. And the result of all this is dillo dirt. Okay?
Very cool.
Great stuff. Everybody ought to be doing it. This is the future. And it started 40 years ago in Austin. And I take particular pleasure in thinking that even though this plant is kind of far north, and I don't know whether the waste from our house would make it there, I like to think that maybe I'm recycling a bit of myself into my garden.
Nice.
There you go.
So I have two comments to make. One is that I think we should do another TWiV in Austin and take a field trip to this place.
Oh,
Yes.
Yeah,
what a good idea.
Yeah. And then the second one is that on the link that you gave, it's just words. So I clicked through to the first thing that isn't there, and it's a picture of the Hornsby Bend Biosolids Management Plant. And then if you scroll down, there's a picture of these birds on this branch, and then in between them are all the baby birds. So pretty. Really cute.
There's, if you look further down in that document, there is a report on the sales of dillo dirt. That ends in 2017. So this hasn't been updated for a while, I would guess. But there is a disappointing trend. Where from 2007 to about, maybe 2010, it's either steady or increasing. And then there's a steady decrease from there to 2017. And I don't get it. This is great stuff. Go out there and buy dillo dirt. I got 4 bags sitting in my garage that I'm not even sure what I'm going to do with yet.
So you did get it, right?
Oh yeah. I bought 4 bags to start with, I think, something like that. I've made about 3 trips for dillo dirt.
Cool. And does it get its name from armadillo, or you don't know?
Some enterprising individual, either enterprising or stoned, just decided we're going to call this dillo dirt. I don't know what the armadillos have to do with it, except that they're prevalent in Austin. Maybe the armadillos hang around the biosolids processing plant.
Dillo dirt. How much is a bag of dillo dirt?
$2. Actually, it may be more than that. It may be more like $4 or something, but I look for the broken ones. They're discounted.
You're looking for a discount on dirt. Okay.
My pick is a.
Wait, we didn't do Alan's.
We didn't do Alan's. I'm sorry, Alan. Alan, what do you have for us today?
So my pick is a music video. It's actually from a concert. I thought of this just recently because one of the performers here just died. Fun fact, a lot of people don't know about me. I used to play the banjo. And so I was certainly aware of Bela Fleck, who's the banjo player in this. He is the master of the instrument. He's the best banjo player since Earl Scruggs, which is meaningful if you know the instrument. And I had heard of the other fellow, Toumani Diabate, who was a master of this instrument called the kora, which is not sure if it's exactly a predecessor of the banjo, but it's they certainly share a common ancestor, and so it's a 21-stringed thing that looks incredibly complicated to play, and this guy is a virtuoso. And this particular performance that I've linked to is Bela Fleck and Toumani Diabate playing dueling banjos from a live show, and you see the versatility of these two instruments and the mastery of these two musicians, and it's just a really neat interplay between the two. And Diabate just died in July, early. He was not old.
So you said you play the banjo?
I used to. Well, I have not played the banjo in quite a few years. If I picked one up today, I would not be very good with it. But one of these days, I'm probably going to get back to that.
All right.
Interesting. I'll have a listen to this.
Yeah.
Me too.
All right. So my pick is an article in Nature. It's photographs. So they have a monthly selection of photographs. But I really, the title is "The World's Most Expensive Dinosaur and More: July's Best Science Images." And so the world's most expensive dinosaur is this Stegosaurus, the most complete ever found. It just was auctioned for $44.6 million at auction. I was watching this auction, and there are people sitting there on their phones because the rich person is not in the room, right? And the auctioneer, this lady, she's just leaning over, and she said 1 million, 2 million, 3 million, 4. It just keep going, and then the guys on the phone or the women would just go like this and this and this. And it went up to 44.6 million. Oh my gosh, 150 million year old fossil.
And
Do we know where it
some
wound
tech
up?
billionaire's obscenely large living room is going to have this in it, right?
No, the person who was anonymous said he wants to loan the specimen to a U.S.
Oh, well, that's
institution.
okay.
Nice.
That's good.
Yeah.
So I don't know who sold it, who had it in the first place, but you can look it up and find the story.
That was a heck of an attic clean out.
It's a beautiful skeleton, right?
Yeah.
Just look at that. Anyway, there are other pictures here, like a lunar cave on the moon. An underground cave on the moon is very cool, and other things. They're all beautiful photographs.
Here's a Lego made of meteorite dust. Okay. These are great pictures.
Yeah.
There's the world's rarest whale found in New Zealand. It looks like it was dead, washed up on the beach. Too bad. They got picked it up in a tractor. But it's very nice pictures. But I like the Stegosaurus. The streets of Arizona. On June 19th, you could see how hot it is. Thermal emissions from Arizona. Yeah. Pretty hot down there.
You'll have to send this to Dixon because otherwise he wouldn't necessarily see this, and he would really appreciate this.
Oh, Lego's made with meteorite dust. Is that the one you were looking at?
Yeah.
Oh my gosh. That's pretty cool. It looks very rough. Anyway, we have a listener pick from Mike. Hello, Vincent and gang, longtime listener. Ran across this article and thought it was worth sharing since it gives a fairly cogent summary of current issues and challenges for science communication, which is what we do, right? We have a lot of challenges. This is from Grow Magazine, which is University of Wisconsin. Science Through a Glass Darkly. The filters of modern news media create disparate, personalized perceptions of science. Researchers at CALS are looking for ways to craft messages that resonate with people of all viewpoints.
Good luck with that.
Obviously, we don't succeed because we have people who don't want to know about the weather.
Really.
Not the leather.
Better stories. Got to tell better stories.
Yeah. Stories are the whole show.
Yeah, it's about stories.
That was my ASV talk.
Yes, which was really good, by the way.
Thank you.
Yes.
Actually, one of my favorite quotes is from Muriel Rukeyser. The universe is made of stories, not of atoms.
Yes.
Don't bother with the rest of the poem.
I screwed up some of this. Hold on. Oh, come on. I put some email behind the picks, which I often do, but we'll do that next time. We do have one other pick from Lisa. I have a suggestion for you after your weekly pick. Listen to NASA's podcast On a Mission, Season 4. Season 4 discusses the Mars rover program. In the 4th season, we'll be transported to the Red Planet with all 5 of NASA's Mars rover. It was fascinating, well written. The guests are interesting to listen to. I enjoyed every one of the episodes. There's no link, but I think you'll be able to find this pretty easily, right?
Yeah.
Paste in a link.
Thank you very much. All right, that'll do it for TWiV 137. You can find the show notes at microbe.tv/TWiV. If you have questions, comments, picks, I'd love to hear from you. Even if you don't like the weather, it's okay. TWiV at microbe.tv. And if you like our work, please consider supporting us, microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
So that's the first time I've said that.
Mm-hmm.
Congratulations
Thank
on
you.
being Emerita. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. I guess we're getting old here. This has been going on for a long time.
Yeah.
Like 15 years?
Something like that.
Yeah.
Mm-hmm.
2008.
Mm-hmm.
Yep.
Alan Dove is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Alan will never be emeritus, right?
No. Yeah. Old journalists don't get that.
I could be emeritus sometime, but I
Yeah.
still have an appointment at Columbia. I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.